let's jump into it, okay? Okay. So what we'll be talking about in chapter 3 are the main methods that microbiologists use to study microbes. And starts right off the bat with what's called the 6 I's. And you'll need to know these 6 I's. You'll need to know them probably by Friday is what I'm thinking. All right, wink. Here's a table. You don't need to memorize the table, but these are the 6 I's: inoculation, incubation, isolation, inspection, information gathering, and identification. And I'm going to go through each of these subsequently and give you a little bit of a description of what that is. All right. It all starts off with the collection of a sample. All right. Whether or not you're an agricultural microbiologist like we talked about, or a microbial ecologist that wants to see what's living in the soil or the water, or in extreme environments like icebergs and volcanoes, you're going to need a sample. For most of you, it's more relevant if you go into nursing and PA and stuff like that school. You will need to have samples from a patient that comes in and is presenting with some kind of discomfort. And so maybe they provide a urine sample, or maybe the nasopharyngeal brain tickler swab, or a blood sample, right? And then that goes off to the lab, and they do these 6 I's with it, basically, right? But the first step is always getting your sample. Once you have your sample, the first step is inoculation. So you'll take that sample, a little bit of it, and you'll put it in something or on something that is packed with nutrients that the microbes like and will allow it to grow. Okay, so this is the step you guys did last week when you had your plates and you put your thumb washed and then your thumb unwashed, and you coughed and you did your swab of your phone. You inoculated the plate. Okay, it can be done on a plate like you guys did. You can also inoculate broth, so a liquid. It's essentially broth, right? And they call it broth because back in the early days there weren't these scientific reagents. It was microbiologists just saying, well, what kind of things grow stuff? And their leftover stew, if they didn't refrigerate it, which back then they didn't have refrigerators, it would grow mold and bacteria. So they would use soup broth and beef broth and stuff like that to grow it up. And that's what we still use to this day. It's a little bit more modified, but so you'll inoculate on agar or in liquid, and then the next step is to incubate. Oftentimes when we're looking at microbes that are going to be in a human, right, you'll incubate that at body temperature, so 37 degrees Celsius. You guys did your incubation step at room temperature, okay? So it just depends on what you're trying to grow up and what equipment you have available. This incubation period can be a matter of hours or a matter of days. If we grow something at 37 degrees, that really increases the rate of cellular division. And so anywhere from 1 to 2 days is pretty standard. By 48 hours, you can see your culture growing. Now, you guys, since you were incubating it around room temperature, the idea is that it would go for a week, 7 days, because it doesn't grow as quickly at room temperature. So inoculation, incubation, next step is isolation, okay? So you'll take that culture, your liquid broth that's no longer clear, it's now cloudy because it has a bunch of bacterial cells in it, or you'll take your plate that you inoculated, and you'll take a little loop, which is a little tool, I think they're showing right here, and you'll you'll take a little bit of the sample and then you will spread it on another plate, or you'll put that sample in another tube. And the idea is that you isolate your microbe of interest from all the other microbes, because remember, microbes are ubiquitous, they're everywhere. And so if you take a urine sample, it may not just be one microbe in that sample, right? If you take a blood sample, it may not just be one microbe. Certainly, if you're looking at volcanoes and icebergs, there's going to be lots of different microbes in that. So you need to isolate them. And how you do that is in one way, you can streak it on another plate. And it's more dilute this time. And what you'll get is the presence of colonies. The colonies are these little dots here, okay? And what those little dots are individual assemblies of a particular bacterial strain. Okay, so one cell spreads out on that agar and then it starts dividing. So each little dot on the plate is a collection of clonal bacterial cells. They should all be the same. And so you'll just pick one of those colonies and then start growing that up. And that's how you now have isolated one microbial strain to then follow up to the next step, let me clear this and go back to my mouse. The next step is inspection. So you now look and see what that microbe is. And that's where the microscope comes in, right? Because we can't see these with our eyes. So we'll put a little bit of this culture that we've isolated on a slide, and we'll do some kind of staining technique. And we'll talk about a lot of the staining techniques later on in this lecture. But you're staining it so that you can make it contrast with the background and see what it is. There are other staining techniques that will tell you what type of bacteria it is. If it's gram-positive or gram-negative, will tell you about the bacterial wall that it has. And so you can get some information that way based on these stains. They're called differential stains. Okay, it will tell you either it has it or it doesn't have a particular feature. So that's what our inspection is. We look at it under the microscope, and then we do information gathering. That's the fourth eye. So now we want to say, well, what does this microbe do? Does it produce certain exotoxins? And so we can grow it up and then do assays to see if there are toxins present that are released from the bacteria. Or we can do a DNA analysis here where we want to look if it expresses certain genes or if it has a particular genomic pattern that we're looking for. And so that would be, where's my draw? That would be this thing here, right? So we do PCR, polymerase chain reaction, using primers that bind to a specific part of a genome. And then we amplify it, and then we run it through a gel, and you'll see these bands here, and those bands will correspond with a certain number of nucleotides. And if the band is there, it tells you that that gene, that region, is present in the bacteria or the virus or the fungus, okay? If it's not there, either your PCR failed or that gene isn't present, or the gene is mutated and the primers couldn't bind to it, okay? You can also do immunological tests, right? Let's say you want to see if there are certain antibodies that bind to the bacterial or viral pathogen. So you will coat this dish here with a layer of antibody, and then you'll put your sample on there. And if it binds to the antibody, it will stick, and then you stain it and stuff like that. Or you can do it the reverse, where you put your sample. Usually this is the second way. You put the sample on the plate, your bacterial sample or your microbial sample, and then you add antibody, and you see if the antibody binds to it, and then you stain the antibody. And if it's present, it'll change color. You can look at drug sensitivity, right? So you grow up a culture on a plate, and you put different dots of antibiotics, and you see if you get this, what's called a zone of inhibition. So an area on the plate where the cells die because they get in proximity to that antibiotic. And so they're toast. So you can see some of these have bigger zone of inhibitions than others. And then this has no zone of inhibition. So you say, well, that drug is ineffective on that bacteria. This one is moderately effective here. These guys are the most effective. Okay, so that's our information gathering. We're getting a better understanding of the behavior and the interactions of these microbes. The next step is identification, right? So, identification. What you need to do is you need to figure out what kind of microbe it is based on that information that you gathered. And maybe it's sequencing the genome. Maybe it's just, looking at references, charts, keys, things like that, to eventually take that sample originally that you took and going through the 6 eyes to find out what kind of microbe it is in the end. Okay. All right, that's the 6 eyes. I ran through that kind of quickly, but there's the table at the beginning. And I don't think we'll get to it today, but toward the end of this lecture, they show the use of the 6 eyes in a clinical setting to give you an example. But one of the things that we talked about was inspection, right? And you have to use a microscope. We talked about the microscope very briefly when I mentioned Leeuwenhoek and how he discovered it and invented it. And then you guys had your microscopy Labster simulations last week. Let's talk a little bit about the microscope itself. There are 2 main features of a microscope: magnification, so that you can see stuff that's too small to see with your eyes, and then resolving power or resolution. So making it big and making it clear, right? If it's big and fuzzy, that doesn't help much, right? It needs to be big and it needs to have good resolution so that you can see the features of those microbes. And that's what these microscopes' main function is, okay? So when we talk about magnification, all that is the presence of light and lens, okay? So just like those of you who wear glasses, right? The lens is curved and it bends the light in a certain way. And light is what carries information, right? If there's no light, you're in darkness, you can't see. There's no information without light. Light carries the information and it carries it through the lens, which is curved, and that allows the light to bend in a way that magnifies it and makes it enlarged so that we can see it. Here's the anatomy of a microscope. And, unfortunately this quarter, if it was face-to-face labs, you guys would be using these microscopes and getting very familiar with them. But as such, we won't be doing that. So I'm not going to belabor the anatomy of the microscope, but I'll just tell you a little bit about it so you know how it works. Okay, it starts off, you have a base, which is the bottom, right? Bottom of the microscope. And then you have this light source underneath, okay? The light source is just a bulb. And the bulb is going to send light up through a filter, okay, which is this thing right here. And then through that filter, it'll go up through this iris diaphragm, and then through a condenser. And then finally, it will hit your sample, your slide that you have your sample on. And what's the point of all these things? Well, you want to, when light is present, it scatters everywhere, okay? What we want to do is we want to take that scattered light and we want to focus it so that all the light waves are going in the same direction, so that that is what gives us our magnification and our resolution, all right? Kind of like for those of you who can't see well, if you can't see, you don't have your glasses, you squint, right? Why do you squint? Because you're closing you're closing down your aperture, okay? And you're letting all that aberrant light, you're filtering it out, okay? We do the same thing with the microscope. You close down that iris, sorry, there, we line it up there. You close it down and it lets that aberrant light that is going in different directions to not be a part of what's going into your sample. And by doing that, we get resolution magnification. From there, it's going to hit this objective, and that's got the lens, the first lens, that's going to magnify the image. From there, it goes into the ocular lens. This is where you put your eyes, right? And you shouldn't make the mistake of just walking up and putting one eye. You know, if there are two, you got to put two eyes there. And you can adjust these. There's a lens in here also. So magnification is made by the objective lens and the ocular lens, and those will combine to give you an overall magnification. And of course, you use these focus knobs. There's a coarse knob to move it, you know, up and down in the Z position, and then a fine adjustment to really get it in focus. All right. Now, by doing all this, by using this light and passing it through these lenses, it will actually invert the image, okay? So as, if you look over here, as the light is passing through this anatomy of the microscope, it's going to take the real image and it's going to flip it like this, sideways. And then when it goes through the ocular lens, it's going to flip it again vertically so that it looks like this. And there's a little test that we do, like when we have the first micro lab and you're using microscopes, you put a little letter under the microscope and it's an E, and you put the E under there in the right orientation, but when you look at it under the microscope, it's flipped to the side and then flipped upside down. And that's just a process of the lenses doing that, okay? So, you know, you don't really have to worry much about the microscope because, again, we're not going to be using it in this quarter, but you should be somewhat familiar with it because these are the most powerful tool in microbiology. So, any questions on that so far? No? Okay. Oops, what happened? What did I do? I think there we go. So that, again, the objective lens forms the real image, which, like I said, it flips it sideways. The real image is then projected onto the ocular, and that becomes the virtual image, which is then flipped upside down, okay? Total magnification. So what you see with your eyes when you look into a microscope is going to be the combination of the objective power and the ocular power, right? The things, the oculars that you look through. So if you have a 40x objective and a 10x ocular, what's the overall magnification?
400. 400.
Exactly. 400. If I said you have a 10x ocular and you want to get 2000 magnification, what lens would you have to use? What would you say? Or what objective would you have to use? You would say, what?
What was the starting again?
You have a 10x ocular, so the eyepiece, and you want a total magnification of 2000x. What objective power would you need?
200.
200.
Exactly. All right, hold on. Just a second. Boys, Zoom. Okay. All right, basic, right? Pretty simple. Resolution, right? So magnification is making it bigger, but resolution is what makes it clear. The resolution allows you to see, for instance, let's say you were looking at something like germs on a hand, and if you had poor resolution, you might be able to see objects that are this size, okay? So then it makes what you see overall look like this. You can make that out. That kind of looks like a hand, right? But if you had much more ability to focus and get those, go from seeing this to seeing something like this with a bunch of different particles in that area, now you get better distinction and you get overall better resolution, right? So pretty straightforward. Resolution makes things clear. It makes you see structures that you wouldn't be able to see normally if you didn't have good resolution. And the better the microscope, the better the resolution, typically. The cheaper microscopes, they can magnify it, but it's not as crisp. How do we make resolution? We take the wavelength of light and we divide it by 2 times the numerical aperture of the objective, okay? What does that mean? Well, so we're looking at visible light, right? Because we have to be able to see it. And that's going to be a wavelength of 400 to 750 nanometers. So that goes on top. And then 2 times the numerical aperture, or the NA of an objective. And if you were to look at a microscope objective, you would often see on that objective, it would say NA, and then it would give you a number. And that's typically from 0.1 to 1.25. And it's really a non-value number per se. What a numerical aperture is the way that it refracts the light, the way that it bends the light, and the degree that it bends the light, okay? So if you have a shorter wavelength and a larger numerical aperture, you will have better resolution, right? Because the overall number, the overall RP, or resolving power, is going to be higher. So something close, oops, sorry, go back. So something closer to this range on the light and something closer to this range on the NA is going to give you better resolution. Now, another way that we can kind of cheat it is to use immersion oil, okay? Immersion oil. Why do we use immersion oil? Well, I'm going to tell you right now why we do that. Let's see. Air has a different refractive index than glass, all right? What that means is air bends light in a different way than glass, okay? And the light in this microscope is going through air and then going through glass and then going through air and then going through glass and so on and so forth. And so what's happening is that light is bending each time it goes from a different media with a different refractive index. And so the example that someone taught me a long time ago is that, let's say you're rollerblading and you're rollerblading on pavement and you're going at an angle toward grass. Once you hit the grass, if you're going at an angle, one of your rollerblades is going to hit the grass first, and it's going to slow down, right? Because your rollerblades aren't going to go as fast on grass as they will on pavement. So you're going at an angle, the one rollerblade hits the grass and slows down, the other one is still going fast because it's still on the pavement, and now it's going to change your overall trajectory. You follow me? Does that make sense? A little bit. Same thing with light, okay? When light goes from one refractive index to another, it bends. That's why when you look at a straw in a glass cup, it looks like it's bent, right? Because you're looking at the straw, and the light that is information is going from air to glass to water with 3 different refractive indices, and so it bends the image. If you are spearfishing, what do? Do you throw it right at the fish? No, you throw it right in front of the fish, because where that fish is in the water from your line of sight is actually off a little bit, because the light that is information is bent because it's going from air to water. Okay, so why do we use oil? We put oil, so if this is, let me get back to my annotation here. If this is your slide here, and this is your lens here, the light is going to go through the glass slide with one refractive index, and it's going to hit air, and it's going to bend, okay? And you're going to lose light. You're going to have stuff bounce back down. You're going to have stuff go this direction here, and you're not going to collect it all. So you're going to lose information. But if you put a drop of oil that has the same refractive index as glass, and they've designed it that way, they've found oils that have the same refractive index as glass, then the light goes through the glass slide, goes through the oil that has the same refractive index, so it's not going to bend the light, and then it goes through glass again, and all these chicken scratches are hopefully showing you that it doesn't bend the light as much. It gives you more light. It gives you better resolution. Make sense? Okay. That's why we use immersion oil. Now, there are various different techniques for microscopy, and we'll go through those now. Bright field is the most common. That's typical. When you come up to the lab and you get a microscope, you just turn the light on, and what you see is what you get. You see a bunch of light, and oftentimes the sample might be a little bit darker, especially around the edges of the cell where the light is again refracted, and so you don't see it, right? So you see a little bit of structure. It's not much though, right? It's kind of hard to distinguish, and but it's cheap. You don't need special filters. You don't need to do any stains of any type, and you can use it for live and preserved specimens. Okay, that's bright field. Now, dark field is the opposite. Okay, dark field is where you have a brightly illuminated specimen surrounded by a dark field, hence the name. Okay, so bright field, everything is illuminated, including the field. What we call the field is the background. Okay, so this is like a soccer field, and all these cells are the soccer players. The field is bright also. But in dark field, it's black. And that allows things that are stained or that refract light well to be visible and to come out of the background. And again, this can be used for live and unstained specimens. Okay, so all these techniques are going to have different advantages. If you just want to do a quick analysis, you don't want to take the time to fix the sample, stain the sample, rinse the sample, cover slip the sample, let it dry, then these techniques work a little bit better. Phase contrast is a technique where you put in a filter into the microscope that will essentially modulate the light waves that are passing through the sample, okay? And so what it does is, rather than having Oh man, I gotta draw. I am not a good, I don't draw well at all. But I particularly draw poorly with a mouse pad, so forgive me. So here's your light wave, right? Terrible light wave. And then you got another light wave like this. Right, that they are in phase with each other, okay? Because they have this, their troughs and their peaks are at the same position. What phase contrast does is it takes the light waves and it offsets it. So it looks like this. Okay. And by doing that, it affects what you see. Okay. It changes the light intensity. And so it allows you to see certain structures, like intracellular structures, particularly with higher resolution or better contrast. All right. So if we compared this to the just the bright field, oh, come on. Bright field, everything's illuminated. You can kind of see some things here, but they don't stand out that much. If you modulate the light waves so that they're contrasting with each other, you get this, and structures pop out a little bit more. Again, this is good for live specimen, okay? Or things that are unstained. We'll talk about stain in just a minute, but things that aren't stained so that they have a color which helps them stand out. Fluorescence microscopy. You probably dealt with some of this in the confocal microscopy simulation, but essentially what you're doing is you're staining your bacteria with a fluorescent dye, or you're using antibodies to bind to certain proteins on the specimen, and then you have little tiny fluorophores tacked on to the end of those antibodies that will fluoresce. And so you then bombard that with fluorescent light, and you get fluorescence, and you have a filter that collects it, and you see something, you know, like this down here. I don't know why they use that image from the book, because that's a really kind of terrible, that has terrible resolution. So it's not the best example of what something should look like. But this would be good, for instance, in diagnosing infections, because you can have specific antibodies bind, and based on that antibody, you can tell whether or not it's this type of virus or that type of virus or this type of bacterium that have specific proteins to that species that the antibodies will target. Confocal, which you guys did in the lab simulation, instead of just shining it with fluorescent light, you take a laser, usually a helium neon laser or an argon laser, and the laser scans the specimen. So it doesn't just flash on the entire specimen and collect all that data. What it does is it has a little tiny pinhole, and that laser goes through the pinhole, and then it scans the image, okay? So you're not seeing it in real time. It has this pinhole that goes back and forth, left to right and then down. And then what it does is it composites that image on the computer screen, and you can see it kind of like unfold. I don't know if you guys are old enough to remember dial-up, but when the internet was dial-up, you'd click on a picture or something or a website, and it would just slowly start to materialize, right? Because internet was so slow. It's kind of like that. You see that image start forming on your computer screen. This gives you a lot better resolution because you're not collecting all this ancillary light information. You're just getting a pinhole that's scanning and filtering out all of the noise. So confocal microscopes are very expensive, and they're not that common in just any type of lab, but their advantage is if you want to get an image, a high resolution image you use confocal microscopy. If you just use fluorescence microscopy, sometimes you can get images that look like that, which are blurry. This is much better, right? Okay. This is actually, well, this is so I did a lot of confocal microscopy over the years. And so this is an image that I took of a worm, a little tiny microscopic worm, and the red is an antibody that labels their intestine. And the green is an antibody that labels the virus that's infecting that intestinal cell. And what's cool about confocal is that you can also see that yellow band right there. That's where the green and the red are co-localizing. And so in light optics, red and green make yellow. And so that's where you can see that. So you can also look at where two fluorophores are localized in the same area of a sample. Electron microscopy is what you do if you need to magnify it way, way more than your typical microscope, okay? And what they do is they manipulate the electrons so that they travel in wave-like patterns. And so this allows them to have a much lower wavelength than the visible light wavelength spectrum. Okay, remember what we said about resolution? The lower the wavelength and the better the NA, the higher the NA, the better the resolving power. So these electrons kind of serve as light waves, only at a much lower nanometer frequency. And so that increases the resolving power. And then that's what you need for viruses and smaller things. Here's a list. You don't have to memorize this, of course. It's just here to give you kind of the differences between these techniques, okay? Transmission electron microscopy, that's, again, using electron microscopes, and you're honing in on the sample, and you're collecting everything that bounces back, okay? The difference between transmission electron microscopy and scanning electron microscopy is that it works similarly to confocal, where you scan the image and you reassemble all that information into pixels, and you get a higher resolution. Gosh, why does it keep, there we go. You get a higher resolution, okay? So you get this here. You can see the structures. It's kind of fuzzy, right? But scanning electron microscopy gives you much better resolution. All right. And those are the main differences that will be helpful to know. When we make our samples, we can do what are called wet mounts and hanging drop mounts, where you just put the live cells on a slide and put a cover slip on it, and then you look at it. So if you want to see if they have things like flagella, that they can move around in the water, if you want to look at their shape, because if you fix them, sometimes that will cause them to dry up a little bit, and then their morphology is compromised to a certain extent. So wet mounts are good because they give you a view of what the microbe looks like and how they behave without being processed. Fixed mounts, of course, are when typically you heat the specimen, so you kill it, and then that helps it stick to the slide, and then that's called a smear. Okay, and then you stain it with certain dyes to look at the presence or the absence of different structures. So each have their benefits. You can't look at a bacterium in real time. If you fixed it, right, it's killed. But then sometimes it's hard to see wet mounts because you don't have the contrast because they aren't stained. We have different types of dyes. We have basic dyes, and then we have positive staining, okay? So a basic dye is a positively charged chromophore. A chromophore is just a the stain, what makes it, what gives it the color or the pigment. Okay, so it's positively charged, and then it's going to bind to things on the specimen that are negatively charged, hence the cationic. Positive staining, it stains the surfaces of the microbes that are negatively charged and attract the basic dyes. Okay, so that's basic dye, positive staining. The dye is positively charged, it attracts the negatively charged parts of the microbes and stains those things. So here's your slide here. Here's a microbe and it stains blue. And there might be a little bit of blue background from the stain if it wasn't washed off all the way, but it's going to stain the actual specimen itself. Whereas negative staining stains the background, not the sample. Okay? So it's anionic, and it uses acidic dyes, not basic dyes. So it's a negatively charged stain or chromophore, and it's going to be repelled by those negatively charged structures on the bacterium. And so you're able to see your specimens because everything else, the field, has been stained. Make sense? So there are two ways. You can stain the sample or you can stain the background. One's positive staining, one's negative staining. That would be good to know. Simple stains are when you just use one dye. Differential stains are usually using a couple different dyes. So you'll use a primary stain to get certain structures, and then you'll counter stain, meaning you'll put a second dye on there to distinguish the cell type. So when we do gram stain, there's a gram stain Labster. The gram stain is kind of the quintessential stain in microbiology for bacteria. You'll learn about things. You've probably heard about things like gram positive, gram negative bacteria. That's what this stain does. Acid fast. And so they'll take up that dye and they'll turn that color, or they won't take up that dye. And then based on that, you can differentiate what kind of bacterial structures it has, hence the differential stains. Okay. Structural stains are going to particularly give you information on the presence or absence of different structures in the bacteria. So a capsule is like a thick coat around a bacterial cell. Not all of them have these capsules. It's like a protective barrier. And so if you use something like India ink, truly just really dark ink, and you put it on the bacteria, it will form this little halo. You'll see a black background where all the ink is, and then you'll see these halos where the ink can't penetrate into that bacterial cell because it has a capsule, a thick like shell on the outside. There are also flagellar stains. So a flagellum, we'll talk about, it's like a little tail on the bacteria that allows it to move around. Not all bacterial cells have flagella. So if you have a stain that binds specifically to that, you can see it and you can distinguish it that way. So here's just, again, examples of simple and negative stains. A simple stain is one dye. So methylene blue would be an example of that. It's just blue. It stains the cells, and you might see a little bit of blue in the background because it binds a little bit to the glass slide, especially if it's charged. It stains the cell. Negative stain stains the background, right? So it shows the contrast of the organism is that it's white on a black background. Differential stains, for instance, this is the gram stain. If it stains purple, that tells you if it's gram positive or negative. If it stains this pinkish, that tells you again if it's positive or negative. And when we go into the gram stain in the next chapter, well, in I guess we're going to skip over that. We're going to do that in a couple weeks. You'll know exactly the steps that you do to do these stains and what they mean. It also gives you information, like for instance, these are rods. So this is a bacillus bacterium. It's not circular, it's not spiral. So it can also give you morphological features in addition. But it's differential because it's either this or that. The methylene blue is not, right? It just stains all cells present. It can't tell you if it's gram positive or gram negative, which tells you a little bit about the gram positive and gram negative tells you about the bacterial membrane. This doesn't tell you anything about that. It can tell you the shape, but that's about it. Here's an acid fast stain. Again, it's going to stain certain cells red, and it's going to stain certain cells blue, which is kind of hard to see. I think, you know, that right there is a blue cell. Most of these are red, but there's some blue in there. And based on being acid fast or not, gives you information on the biology of that bacteria. Another one, spore staining. So the green shows the presence of spores, and red shows vegetable cells, or basically just non-spores. So just a typical bacterial cell. So again, it differentiates. Is it a spore or is it just a regular cell? That's the beauty of differential stains. A structural stain, this is that capsule stain. So you put India ink on it, and then now you can see these like halos here. That's the bacterium right there. And the halo is that capsule, that protective shell that the ink can't penetrate. And so that says, oh, okay, well, all of these bacteria have capsules. If you didn't see these halos, then you would know that there isn't a capsule there. Flagella. So here are the little flagella, these little squiggly things coming off of this bacterial cell. And you see not all of them have it. Just, you know, this one doesn't, that one doesn't, some of them do. Structural stains. Fluorescent stain, right? Here we're looking at a chromosome in green, so that the DNA in the middle stained green, and the membrane stained red. And so that gives you, they've used antibodies that bind specifically to the membrane receptors. And they probably use some kind of dye that intercalates into the DNA, into the nucleotide base pairing structure of bacteria. And that elucidates the different features and where they are in your specimen. Okay, that's it. We're out of time. The rest of this slide deck, like I said, goes into like a case example of the 6 eyes in a little bit more detail, but we went over this, so I'll let you guys review this, the rest of this, if you want to, on your own. For the quiz on Friday, I would focus on what we talked about today, okay? Hint, hint. So the material that we just discussed is what's most likely going to be on the quiz, all right? So are there any, so if you guys want, you're free to leave the Zoom. But if you have any questions on that material, because I know I went over it pretty quickly, I wanted to get it all out there, then I'm happy to stay on for a bit and take questions, okay? And this is recorded, so if you need to go, but you want to see if there were any questions that were relevant, then you can check the video out later. Or if there are any questions in general, not necessarily about the lecture material, about something else, I'm happy to address that as well. Okay. Well, good. I guess that seems like that was pretty straightforward then, right? Um, I haven't decided what we'll lecture on Friday. Maybe we'll just hop into the next section. Christine, I did update the syllabus schedule. I just basically took out the COVID stuff that whole week, and I'm just gonna, squeeze it in when we talk about viruses. But next week we'll be mostly focusing on host microbe interactions in the immune system. So we'll plan to maybe get into that on Friday a little bit, and then hop into it full bore next week. All right, I think everyone has pretty much signed off, so I will see you guys on Friday.